Welcome. This is chapter 8 of CNP, which is Plant for Drug Discovery. It is a wind-up chapters for CNP of undergraduate students. So today we try to see the plant's role in drug discovery and what type of plants, what type of drug are there that's in the market, in the clinical theory and the preclinical phase. We have to see these very important chapters for you guys, so you have to follow attentively so as to get very important knowledge from this tutorial video. And don't forget to subscribe and share as well as comment on the channel before starting watching the video. Let's go directly to the tutorial video. So the plant as well as plant extracts and inorganic material starts to be used for medicine starting before 1800. However, in 1805, morphine is isolated from the opium tree for the first time, and the structure proposed in 1935 and approved by Synthetic in 1952. Starting from that time onwards, we have to use different drugs that sourced from plants, microorganisms, and different natural sources. So in 1828, you know that the first organic synthesis or urea can be synthesized from inorganic compound, and this statement disproves the science behind organic compound is only isolated from natural product or from living things, because this urea can be synthesized from non-inorganic substance, but it is organic. In 1842 to 1850, the first synthesized organic compound used for medicine like chloroform, dithyramide, especially for anesthesia, they can be isolated from the plant as well, and the synthesized organic compound. This one is. So, example of early synthetic drug include like chloral hydrate, which can be given for sleeping pill or sleep-inducing drug, acetylsalicylic acid or aspirin, that can be used for analgesic drug. It can be synthesized in 1853. The clinical trial started for this drug in 1893. After 40 years later, and the phenazole synthesis, benzocaine, and protonsil can also be the drugs that can be early synthesized drug. And the different drug can be isolated from natural product like quinine, which can be used for anti-malarial, digitoxin, which can be given for heart failure, and the salicylic acid or antipyretic drug can also be started from this drug discovery can be maybe followed from this traditional medicine screening or serendipity drug discovery. This serendipity drug discovery means that discovering a drug or bioactive metabolites through chances. So when we come to the medicinal plant, this medicinal plant can have different bioactive metabolite or phytochemical reserved in their structure in different parts, from starting from their plant, from stem, from barks, from root, and these bioactive metabolite can be extracted first by using different extraction techniques. Then after we have to assess the bioassay technique, whether having the pharmacological effect or not, and the toxicology profile can also be done, and finally the pure compound that can be obtained by fractionation can be checked for their biological activity as well as the structural chemical characterization, so as to know what the structure of that bioactive compound, so as to produce the drug from this pharmacophore as well. So you know that drug discovery is very tedious, and it takes 10 to 50 years and an estimated cost of $1 billion American, and with a probability of a drug will be less than 5%. Therefore, this drug discovery is very, very tedious and very, very expensive, as well as numerous bottleneck. But nowadays we have to start the reverse pharmacology, which is very, very economical, time sparing, and the least bottlenecks of this drug discovery. So we have different scenario of getting a drug with a short period of time and with potent, and the probability of druggable is by far greater than 5%. So the research and discovery path for natural product can start from identifying the traditional medicine. For example, Ayurvedic stage means that this one is a traditional use of medicine in India and China as well. So by that principle, you have to screen whether that bioactive plant or the bioactive metabolite in the plant can be whether it is active or not by using different technique. Then you have to get that bioactive metabolite so as to check it is pharmacological effect on test tube. Then it can be passed to the preclinical phase, especially in laboratory animal. Then if it is very, very promising, we have to bring that bioactive metabolite to what the clinical trial so as to get what the druggable compounds. But this druggable compound can be given still now with different complementary medicine and alternative medicine with different drugs in complementary or in alone. So this is a research and development pathway for natural product drug discovery. So why always we are focusing on natural products? Because living things have relied on nature, and this natural product or the natural source are originated from plants, animals, microorganisms, marine species, and minerals. And these natural products basis of entire human need, including treatment of several diseases, and have a long been of interest to the field of chemistry, biology, medicine, because of their immense molecular diversity, biological activity, and what medicinal properties. Therefore, as we Africans, or when it comes to Ethiopian, we Ethiopian can highly rely on natural products for our healing effect, because they are easily affordable and their biodiversity is very large, and the health sector is very low relative to the number of population in Ethiopia. Due to this reason, we have to highly rely on this traditional medicine. Not only this, the synthetic product is somewhat toxic and lead towards a drug resistance. So finding a new drug that is very important and cannot be developed drug resistance is very important. So the origin of the reservoir of this type of drug is nature or natural products. So these natural products in traditional medicine act as a weapon for traditional doctors. You know that most of the traditional healer does not tell you that whether the plant have to be used for the medicine or not, because they can use it as a weapon for their life, because they get some income from that plant as well. So they can act as what a weapon for these traditional doctors. So natural product in modern medicine as a source of direct drug and the lead compounders profit for pharmaceutical company. So natural product always a source of a new horizon to treat drug resistant disease. So how this natural product act as a drug precursors? So recent interest in drug discovery such as molecular modeling, combinatorial chemistry, and synthetic chemistry can be highly relied on this natural product drug discovery. However, natural products still keep their invaluable roles as a source of medicine, and a natural product source of small molecule drug precursors beyond their direct use in their original forms. And by using semisynthetic approach, we have to increase the yield that obtained from natural product and the high cost of the total synthesis also. So semisynthetic approach can be preferred due to the low yield of natural product and the high cost of total synthesis. So a typical example of drug precursors, for example, if you take 10-decacetyl-basatin-3, a precursor for paclitaxel semisynthesis, you know that paclitaxel can be isolated from Taxus perifolia, but the amount is almost 0.014%, which is very, very low. So to boost the amount of paclitaxel isolated from the plant, we have to use what? 10-decacetyl-basatin-3 as a precursor for paclitaxel to synthesis by using semisynthesis technique. So Holter Research Group developed a semisynthetic method to produce paclitaxel in large amount, and the Bristol Myers Squibb pharmaceutical manufacturer adopted this method in 1992 that of 2002, so as to increase the amount of paclitaxel or Taxol, which can be used for what anti-cancer drug, and this drug can be obtained for the first time from the Taxus perifolia plant for the treatment of what? Cancer disease. Diosgenin also a precursor for progesterone synthesis. You know that diosgenin is a steroidal Topoisomerase obtained from the tubers of various Dioscorea species that grow in Mexico and Central America. And this progesterone is, you know, that the female hormone isolated from ovaries with very low yield, which means that 20 mg per 625 kg of ovary, later synthesized from cholesterol, still with low efficiency, and is a key intermediate for the production of cortisone. So this diosgenin can also be what increased its yield by using the semisynthetic approaches. So this is the structure of diosgenin and the progesterone as well as cortisone. You have to see the structure and have something from this structure as well. Shikimic acid, you know that the precursor for oseltamivir phosphate synthesis. This shikimic acid can be extracted solely from fruits of shikimic tree and Ecilium verum oak, and the latter obtained from the fermentation of genetically occurred Escherichia coli strains, and important biochemical intermediate in plant and microorganisms. Therefore, this oseltamivir phosphate synthesis can be still now, it can be orally active for neuraminidase inhibitors developed for the treatment and prophylaxis of influenza virus A and B. So this oseltamivir phosphate can be obtained from this shikimic acid as a precursor. So shikimic acid can be what the precursor for oseltamivir phosphate. So how these natural products can be act as anti-cancer agent is a very important concept here. Do you know cancer, which is the proliferation of cells? That means, so this cancer is a lethal disease which has deprived 1 million of lives, and it is the second largest cause of death after cardiovascular disease, especially lung cancer, stomach cancer, liver cancer, and breast cancer can be what the first line of cancer disease. As the report of WHO in 2010. So hard to treat due to the recurrence of tumor and as a side effect of chemotherapy of the drug. So this cancer disease can need what searching the drug which have what less side effects and lack of what recurrence of this tumor. So tumor cells are highly sensitive and easily induced drug resistance. So the new anti-cancer drug from natural sources is indispensable option for nowadays. So in the history of anti-cancer research, over 60% of the clinical use of anti-cancer drug originated from this natural product. And this anti-cancer drug can perform their function by different mode of action. And the natural product that targets protein, some natural product can target a protein as a drug target. So faculty regulation of cell cycle or division is one of the most feature of most cancer, and the cell cycle does offer several targets for therapeutic intervention. There's a protein tubulin, Topoisomerase I and the Topoisomerase II are the most important drug targets for anti-cancer. So the protein tubulin exists in both alpha and beta monomers. And during the normal cell cycle, the polymerization into microtubules after mitosis or cell division, and the microtubule dissociate back to the monomer element. So the therapeutic intervention exhibits what assembly or reassembly equilibrium of the monomer or dimers lead to cell death, maybe due to apoptosis or necrosis or atrophy. So this topoisomerase can be mediate the cleavage of DNA single strand, whereas topoisomerase II can mediate the cleavage of double strand. This one is the difference between topo 1 and topo 2, and inhibiting the enzyme via therapeutic intervention is one scenario or one mechanism of action of these drugs. So we have different drugs that inhibit tubulin assemblies. The first drug that affects that inhibit tubulin assembly is vincalcaloid. The drugs are like that of vincristine and vinplastin, which can be obtained from Catharanthus rosas, commonly known as a Madagascar periwinkle, formerly included in the genus of Vinca as Vinca rosas. So Catharanthus rosas is a source of vincristine and vinplastin, which can be used for what? Tubulin inhibitor anti-cancer drug. So isolated in 1950 as a vincalcoplastin and treats Hodgkin disease and the bladder and the breast cancers. But this vincristine can be isolated in 1961-62 as a leucocristine and treats acute lympholytic leukemia as well as lymphomas. And finally, this vincristine and vinplastin inhibit polymerization of the tubulin protein to microtubulin. This one is the mechanism of action, how these two drugs can be performed their effects. Finally, from this vincristine and vinplastin, they produce some semisynthetic analog like vindesin and vinorolipine, and these two drugs can be developed in 1970 as anti-cancer agent, and seems to be more active than vincristine even, and against non-small cell lung cancer, but still it is under evaluation due to its higher hematological toxicity than vincristine, especially this vindesin is very potent, but still hematological toxicity is under evaluation. But vinorolipine can be approved for the treatment of non-small cell lung cancers, and it can be already used. Combretastatin is also another tubulin inhibitor drug. This combretastatin can be obtained from Combretastatin Analogs, a native to tropical and southern Africa, also found in Madagascar, tropical Asia, and tropical America. And this combretastatin can be isolated and still in the clinical phase, preclinical phase, because of can be combretastatin A1, combretastatin A2, and the combretastatin A4 phosphate can be what all the clinical material. That's why you can label it in A1, A2 like this. So this CA or combretastatin A4P approved for fast track phase 2 clinical trial against anaplastic thyroid cancer actually. So have a look how natural product is a miracle source of secondary metabolite which can be used for cancer disease. The compound that promote tubulin assembly like paclitaxel or Taxol, this Taxus brifolia is the source of this Taxol. The Pacificio or Westernio is a conifer native to the Pacific northwest of North America, and it can be known by the trademark known as Taxol. Selected as development of candidate in 1977 based on the good activity against human tumor xenograft in the nude mice. So docetaxel is a semisynthetic paclitaxel analog, and this paclitaxel and docetaxel are now used for the treatment of ovarian cancer, breast cancer, and non-small lung cancer. So this is also another important drug for cancer which can be obtained from plants. Epotilone A to D. Epotilone A to D also obtained from Sorangium cellulosum, a gram-negative bacteria of a group of myxobacteria. Such type of endophile can act as a source of different secondary metabolites. So this epotilone A to D with different structure can be obtained in the phase 2 as well as phase 3 clinical trial, and which can be for the first time actually can be isolated for antifungal agent. However, due to their mechanism of action is similar, now it can be found to have the same mechanism of action with that of paclitaxel. So it is on trial or in the phase 2, the phase 3 trial of cancer disease as well. So have a look how bacteria and the fungi and the algae as well as different marine species can also be what? The source of secondary metabolite, which is very important for medicine. The compound that inhibit topoisomerase I can include from Combretastatin Analogs. This Combretastatin can be obtained from Compoteca cuminata and isolated from Compoteca cuminata in 1966 and entered into clinical trial in 1970 as a sodium salt of carboxylic acid formed by opening the lacto ring, and however due to this water insoluble, no efficiency proper for the development. However, later on they developed semisynthetic analog from this Combretastatin so as to produce what? Anti-cancer analog for Combretastatin. So interest renewed as a primary cellular target was inhibition of topoisomerase I, and these two drugs, topotecan and irinotecan, was a water-soluble derivative of topotecan and irinotecan from Combretastatin. And this topotecan can be in clinical use for second-line treatment of metastatic ovarian cancer and small lung cancer, as well as irinotecan can be in clinical use for the treatment of metastatic colorectal cancer disease. So these Combretastatin are unique pharmacologically in having topoisomerase I as their only target and in being able to penetrate what the mammalian cell readily. Therefore, the different drug can be obtained or isolated from natural products. Maybe that drug have less toxicity as well as have high toxicity. Not only that, may have less water solubility. To solve this, we have to use what? Semisynthetic analog from that drug, which are more potent than the parent molecular compound. So this Rebeccamycin also another drug which can be isolated from catalase positive or gram-positive rod-shaped bacteria, and this Rebeccamycin is an indocarbazole isolated from Nocardia species, and the modification of this indocarbazole skeleton generate a molecule with enhanced topoisomerase I activity, and this fluorine substituted Rebeccamycin analog headed for phase 1 trial for topoisomerase I inhibitors. So this structure PMC 250749 is what the phase 1 clinical trial for topoisomerase I inhibitor drug. So there is also a compound that inhibits topoisomerase II. A best example of this drug is photophyllotoxin. This photophyllotoxin can be obtained from Photophylum pelatatum. It is an herbaceous perennial plant in the family of Barbidaceae and the genus Orinaceae. And this photophyllotoxin was isolated in 1880 from rhizome of American maple and elicited its structure in 1951, showed the inhibitory effect on the formation of the mitotic spindle as early as 1947. So it is too toxic for the use of an anti-cancer agent. But from this photophyllotoxin, they produce an analog which can have less toxic. For example, etoposide and tenoposide also analog of this photophyllotoxin, and this etoposide especially inhibit topoisomerase II used against small cell cancers in combination with cisplatin. Cisplatin also one of the anti-cancer drugs that can be obtained through chances or serendipity, and also effective for the treatment of testicular cancer and a small cell lung cancer as well. Yeah. So this tenoposide also used against a neuroblastoma with cisplatin, acute lymphoblastic leukemia with and small cell lung cancers with carboplatin, and etoposide is also to the circumvent the water insoluble problem associated with etoposide and tenoposide. Actually, this tenoposide and etoposide is less water soluble, but from this less water solubility they solve by producing etoposide, which is highly water soluble actually from the two analogs. So by these three drugs can be obtained from photophyllotoxin, which is obtained from Photophyllum plants and having what less toxic than the parent molecules of the drug. The other one is anthracycline based topoisomerase II inhibitors, and this daunorubicin and doxorubicin also the drug from obtained from Stereochemistry species, and they are anti-cancer or especially anti-breast cancer. And isolated from bacteria of Stereochemistry species used for acute myeloid leukemia, acute lympholytic leukemia, chronic myelogenous leukemia, and so on. So approved for medicinal use in 1979. Hair loss, vomiting, bone marrow suppression, and the inflammation of inside the mouth is some phenomena of the leukemia actually. So this anthracycline is the best topoisomerase inhibitor drug with the other topoisomerase II inhibitors. So compound that interact with other proteins like, for example, geldanamycin. This Streptomyces hygroscopicus can be a source of this geldanamycin, and discovered in Streptomyces hygroscopicus inhibit the function of heat shock protein 90 or HCP90 by binding to the unusual ADP or ATP binding pocket of the protein. So this protein play important role in the regulation of the cell cycle, cell growth, cell survival, important target site for cancer chemotherapy as well. So this also obtained from bacteria, so can very important to us as an anti-cancer agent. Staurosporin is isolated in 1977 from bacterium Streptococcus, and is the first of over 15 alkaloids to be isolated with this type of base indole. Chemical structure is absolutely stereochemical configuration, less dated in 1994, and this UCN01 stereosporin derivative interface to clinical trial against leukemias, lymphomas, and the various solid tumors, melanoma, and the small cell lung cancer that has high binding ability to human plasma protein. So both are non-selective inhibitor of protein kinases actually. So This also another anti-cancer class of drug. So different drugs from natural source can be used for cancer disease. Most of them are in clinical trial, some are in preclinical trial, and some are in the already approved and commercial. So you have to know that how this natural product can be a source of this cancer disease. This is selenosporamide. Selenosporamide is obtained from bacteria belong to the family of Microsporia, Sporacea, and found in marine and sediment across the tropics. And the selenosporamide A can be isolated from totally new genus of Selenospora, identified as a cytotoxic proteasome inhibitor, and entered in phase 1 clinical trial very soon in 2006. So this is also another product that can be isolated from marine species so as to give what healing effect for cancerous disease. Again, some natural product or some non-natural compound that target DNA and RNA. So second major class of anti-cancer agent acts directly on DNA or RNA, either by intercalation, alkylation, or by cleavage of the double-stranded DNA. For example, if you take actinomycin D isolated from Stereochemistry parvulus in the early 1960s and inhibit DNA-dependent RNA synthesis via strong intercalation into the double helix DNA structure, clinical use in the treatment of what? Trophoblastic tumors in females and the metastatic carcinoma of testes, and in Williams tumors in children actually. So this actinomycin D is a DNA intercalating anti-cancer drug. Bleomycin also important classes in the family of glycopeptidolide antibiotics, and this bleomycin A2 originally thought to act as through DNA cleavage and cleave both DNA and RNA in oxidative, sequentially selective, and metal dependent fashion in the presence of oxygen, and recently recognized as transfer RNA inhibitors. So this bleomycin A2 can be act as what? Or recognized as a tRNA inhibitor drug. So mitomycin, especially mitomycin C, is also called mitosins, were discovered in the late 1950s and was approved for clinical use in 1960s. And the only clinical trial used from this used member of this class is mitomycin C, and the alkylate and the cross-linked DNA only after undergoing a 1 electron reduction, and clinically it used for the treatment of gastric and the pancreatic carcinoma. So this mitomycin C is very important for the treatment of gastric or pancreatic carcinoma. Clazimycin, this is also another member of a large class of anti-tumor inidine antibiotics and isolated from Microspora echinospora species. Clazinesis cause a single-stranded and double-stranded DNA cleavage, and too potent and too toxic for direct clinical use, used as a warhead in the antibody-targeted chemotherapeutic agent, and or Mylotarg. Mylotarg was approved for the FDA in 2000 for clinical use for the treatment of what? Acute myelogenous leukemia actually. So this clazimycin is also a very potent, and at the same time it is very toxic for the direct use of clinical use. So it can be used as a warhead in the antibody targeted chemotherapeutic agent. This Mylotarg was approved actually in 2000 by FDA for clinical use of the treatment of acute myelogenous leukemia. So this is also another drug that can be used for anti-cancer drug. Ectinacidicin also a C-squared species in the genus of ectinacidicin, and this ectinacidicin ET743 is isolated originally from Caribbean tanicates. Ectinacidicin carbonate was submitted in 2006 for approval as an anti-sarcoma agent and was recommended for approval in July 2007. The bind to the DNA minor group and alkylates the N2 position of the guanine and inhibits the transcription of specific genes, especially it causes what p53 independent cell cycle block, lead to apoptosis or a programmed cell death. So close to 40% in phase 2 studies on sarcoma. So still this drug is on the trial on phase 2 clinical trial, that means. So it is very important, again another potent anti-cancer drug for the future maybe. Metramycin, it is an anti-tumor. Actually, it can be obtained from Streptomyces palicatus, a bacterial species from the genus of Stereochemistry, and an anti-tumor antibiotic isolated from Streptomyces palicatus, used to limit the extent for the treatment of embryo cell carcinoma of the test and cancer-related hypercalcemia. Specific inhibitors of SP1 transcription factor in hematopoietic cells. This metramycin. Actually, it have 24 stereogenic center, which means that if it have 24 stereogenic center, which means that almost to the power of 24 number of isomers, stereoisomers that it means. So it is very, very large number of stereoisomers that have. Streptozocin, this is Streptomyces acromogens, a species of gram-positive bacteria, and the N-nitrosourea is isolated from Streptomyces acromogens and acts as actually a DNA alkylating agent and recommended for the use in combination with doxorubicin as a drug of choice for the chemotherapy for patients with malignant neuroendocrine pancreatic tumors. So this one is given for in combination therapy with doxorubicin for the treatment of malignant neuroendocrine pancreatic tumors. That is it. So the next topic is enhancement of natural product as anti-cancer by using what technology. Do you know the limitation, the use of a single chemotherapeutic drug as anti-cancer agent are emergence of drug resistance, high cell toxicity, limited regimen of clinical use, poor drug solubility, instability of active form in vivo, and the cell toxicity are there. So the need for new chemotherapy, new therapeutic target to improve the efficiency of natural compounds to reduce toxicity as well as side effects, increase selectivity, and reduce the risk of using chemical medicine alone is very risky. So the methods such as polymeric nanoparticles, combined chemotherapy, combined immune in the chemotherapy, flavonoid metal complex are very promising technique for the future, which can be using by technology so as to reduce the risk and increase the benefit from this natural product, because they are very potent reservoir of this anti-cancer drug. The last topic for this chapter is the home tech message. You know that these natural products have served historically as a major source of direct drug and drug precursors employed as starting material. So natural product can enhance immune cell as well as body immunity, anti-tumor effect, and inhibit the proliferation of the cancer cell. So this anti-cancer research begin to focus for looking for high selectivity, strong activity, low side effect, and wide range of anti-tumor drugs from natural sources. Not only biologically toxic compound, but also more non-toxic ingredients have attracted people attention in anti-cancer agent, and the development of cancer prevention and the healthcare product from natural product has a broader prospect and a greater economic and social benefit when compared with synthetic anti-cancer drug. It is also a great alarm or homework for all stakeholders in this area, as there are undoubtedly more unique and potent biologically active natural products waiting to be discovered. So this home tech message can be given for every student who read or who watch this video. As far as you have seen different natural products used as anti-cancer, and this natural product drug discovery can be originated from plants, from microorganisms, or from marine species. So you have to scan your environment so as to get a very, very potent with less side effects or less toxicity drug from this natural product. So it is a home tech alarm for us to search what, to scan our environment so as to get what very potent anti-cancer drug for this very, very dangerous disease in our country or in the world as well. So generally this one is all about the topic. Thank you for watching, and you have to answer this quiz question by yourselves, and you have to write the answer on the comment section of the our channels, and the first three person who answered this question correctly, especially question number 1, question number 2, question number 3, the question number 4 only, leave about question number 4. If you answer the question correctly in the first list, the 3 first the audience can be what get the mobile air card. So you have to use your chance by writing your answer on the comment section of the channels. Generally, thank you for watching our interesting tutorial video, and don't forget to share, and don't forget to subscribe, as well as don't forget to like the channel, so as to get what very important tutorial video for you. So thank you.